Good afternoon and good morning, good evening, good night, wherever you are in the world. We welcome you to our ISSS ISTA webinar. You see Ruth here, who is presenting one topic. We're still having difficulties with Luna to get online a little bit, but we are solving that in a few minutes. We have Julia here as the president of ISSS, and I welcome you to this webinar series now. The name today of the webinar is Seed Pathology, the good and the bad microbes. And that's like always in life, there are good things and bad things. And they may be all the same or very similar or the same family or whatever, but one is helpful and the other one is less helpful, I would say. And sometimes you call that good and bad. Before we start, I hand over to Julia to say a few words as the president of ISSS and maybe getting you some more information, invitations. Julia, the floor is yours.
Okay, thank you very much, Andreas. So just before I show the little information from our society, I just would like on behalf of the International Society of Seed Science to extend my heartfelt congratulations to the ISTA for hitting the incredible milestone of 100 years. I think this really highlights the central role of seed quality in everyday life and the importance of your society. And with our society just being 25 years old, well, we're kind of the new kids on the block compared to your impressive history. And I hope that our collaboration will continue like we do in the form of these webinars, which really show how our communities are connecting together. Okay, with all of that, I just would like to show you a screen. Yes, it's working. Okay, so just 2 slides on the International Society for Seed Science, because we had some recent changes that I wanted to share with all of you. There's been a main difference in the way we deal with memberships. So memberships are not very expensive in our society, £30 for members and then £10 for students. But just to let you all know that we have a membership waiver for low and lower middle income countries. So please join us. It gives you access to a lot of information, newsletters, and I would also like to suggest promote our social networks via LinkedIn, via the International Seed Science Society, via X, where you'll find a lot of different posts on our journal, on the publications from our society's journal, people asking for help, asking for information, and more information on what's organized in relation to seed science. And just the last word in terms of our journal, Seed Science Research, there will be it's open access now. There will be a new issue on seed innovation systems from our previous biannual meeting that will be coming out in July. And there is a request for publications on methods and new innovations in seed science. So if you're interested, make a proposition to the editor. You can find the information on the website. And there as well, for lower middle income countries, there are a lot of possibilities to get waivers for publications fees. So please check it out. So with that, just one last slide to introduce you to the place where will be held the next in ISSS meeting. It will be in Kings Park and Botanic Garden in Perth in Western Australia. And it will actually be a joint meeting of the biannual ISSS conference, the general meeting, and the 8th Seed Ecology meeting. So it's from the 15th to the 19th of September in 2025, and will be organized amongst others by Dave Merritt. And with that, I would like to, first of all, I would like to take this opportunity to thank both of our speakers today for all the efforts that they put in giving us a presentation. And I would like to thank the organizers and the ISTA for organizing this webinar. And I give the hand back to you, Andreas.
Yeah, thank you very much, Julia. As everybody is aware, probably who was in our previous webinars, this is recorded and will be made available by all both organizations later on, and you will be able to review it, for instance, on the ISTA YouTube channel, but I think there's also a possibility in the channel of ISSS. What I would like to remind you is that if you have questions, please put them in the question box, and we will try to answer them. If they are not answered today, they will be answered at a later stage. And you will find both the presentations which you see today also on the ISTA website. You got the link, and you can find the presentations there. Now I would like to introduce Ruth a little bit. Ruth Barnhorn. Ruth, as you may know, I'm not very far away from you, less far than normally. In Rotterdam at the ISF Congress in the moment. Ruth is working very close to here at Nactambo in the Netherlands. Ruth, don't let me say the name of the city where you are located. That is too complicated for me. You work there as a scientist in vegetable seed pathology. You are a specialist in that and a member of the ISF, International Seed Federation, International Seed Health Initiative for Vegetable Crops. That's ISSA-WEDGE, and that's since 2015. And in 2019, you became a member of the ISTA Seed Health Committee, and you are currently chairing this committee. And we thank you for that, for instance. And your daily work consists of development and validation of tests, methods, and protocols to be used for the detection of all kinds of pests being harmful or not, or on or to plant seeds. I think that is an important task, and that's also what we would like to hear from you. And your title is The Incredible World of Seed Pathology and How It's Used to Prevent the Next Plant Pandemic. If you put your display mode totally, then we are able to see everything. I think you have to go again into display settings up there and make it duplicate slideshow, and then it should be working. That's it. Ruth, the floor is yours.
Ruth, we don't hear you.
Oh, sorry. I was on mute. Sorry for that. Well, Andreas, thank you very much for this nice introduction. And thank you all participants being present and willing to listen to my talk. Indeed, it will be on the seed health work that is my daily work, and I hope to educate a little bit more on what seed health is about, and also what ISTA is roles in seed health is. Okay, so seed health, seed health has to do, of course, with pests being present on seeds, and I'm dealing it. Wait a minute, one moment. I'm getting rid of these things that are in the way. Okay, so well, back in the 1970s, in the late 1970s, a study was done on the investigation on transmission of pathogens via seed, and due to the increase of trade, but also, of course, trades that were already there for years, and pests appearing in different countries around the globe. So making these connections, as you can see here in the picture, there was a scheme drawn by the scientists, which name I couldn't really trace back. But well, the investigation went on to 25 pathogens, commonly known as on seeds, and how they were introduced or traveled around the globe, and caused the emergence of disease via the pest dissemination. So this was actually a big start of seed health initiatives coming around the globe, and had the focus on pests on and in seeds, being a major risk for yeah, food production in the end. Okay. So, well, seeds, of course, what kind of pests are present on seeds? And well, you can think of fungi pests, of course, like Botrytis cinerea or Leptosphaeria maculans. But also bacteria, but different bacteria are able to be present in seeds, causing risks, like different Xanthomonases, Brassica and carrot here as an example. Viruses, of course, can be a major issue on seeds. CGMMV, cucumber moth, and squash mosaic virus are examples of that. Nematodes, or little worms that we cannot see with the eye, that can also infest seeds and travel with these seeds, as do viroids. Being a more recent pest group that was discovered, and being a big threat, consisting of an RNA strand that yeah, causes virus-like infections, and also known, but quite new actually in seed health regulations, are insects that well, can harvest seeds, as you can see over here. Harbor seeds, I have to say, and lay their eggs in seeds, and yeah, being very well, being able to be to create a lot of diseases worldwide actually, and spread easily via seeds as well. So there's quite a lot of different pest types that are yeah, considered to be a risk for seeds traveling the world via trade. So what is the location of these seeds? So you have seed-borne pests. So seeds pests that can, yeah, go out of the seeds or come from seeds and cause a disease spread. They can be carried externally, of course. So yeah, the outside of the seeds, the seed surface. As you can see here on the low bottom, you see yeah, a tomato seed with red dots outside of the seeds, being the pathogens present inside the seeds, but also occasionally, and even a bigger problem actually, is pests being present inside the seeds, and that can be in the different layers of the seeds, seed coats or endosperms, for instance. And due to the fact that they are more embedded into the seed, the colonization is more deep, of course, considered to the fact that they are outside of the seed stick to the seed surface, the impact is can be yeah, far much more devastating, since seed treatments that we currently do or are less effective, and detection methods as well. Well, you have to crush the seeds or grind seeds even to get the pathogen out of the seeds and try to for the detection of these pests. So the seed pathway, the entry and the establishments in new areas, yeah, is done. That seed is of course a pathway then, and it's a potential of emerging of the diseases. And you have seed-borne pests that can be seed transmitted. These can be pre-emergent, so can be done in the soil, that they infect the soil actually, but also when the seeds actually show seedlings. And then when the seedling appears above the ground, the pathogen goes into the plant or spreads into the plant, and in a later stage can infect other plants yeah, growing next to these infested plants. Well, here are some examples of actually fungi that yeah, come from the seeds. In this case, you cannot see whether they are from inside or outside of the seeds position. But what you definitely can see in these pictures is the spread event. So here you see in the second picture, you see some seedling coming out of the seeds, but also a fungi is growing to neighboring seeds, and this is done on media in this case, a standard tests that we do to see whether the presence of the pests are there, yes or no. But pests can also escort seeds independently, and that's not directly detached to the seeds itself, but or mixed with debris. But it's also, but still has a potential to contaminate the environment, and that is a big issue. And therefore, the World Trade Organization came up with an international standard for phytosanitary measures to prevent, with all kinds of rules and with descriptions on how to treat your seeds in such a manner that the risk that it is spread via trade is minimal. And one of these chapters inside this standard is, of course, the testing for these for pests itself. Others are treatments, of course, is also a chapter in there. So why do we test for seed health? Well, of course, you gain knowledge from the seed parties that you're testing, and you can also by doing this check whether you should go for treatment and what kind of treatment. If, for instance, hot water, zinc or hot water treatment is done, or hypochlorite, more chemical treatment is done. But also, if you know that your seed parties are infected, then you should not trade it, of course, and then you prevent the introduction of the pathogen in new regions. And that's helpful, of course, for governments, but also for seed companies, because governments also because, of course, that they don't want to have these pests entering their borders and going to be prevalent in the end. But for seed companies, it's the other way around, of course, because if you sell a seed batch which is infected with a certain pest and causes an outbreak, then you can already think about the fact that there's going to be claims for the company. And for the producers as well, because if you have an open field and you sow a pest-infested seed batch, then it might end up that you cannot use your area for a couple of years for that kind of crop. So that's all very important. And in the end, it's a guarantee for the customers that these seeds are healthy. Well, there is a regulatory framework that comes with this, and that's one is the IPPS ISM movement of seeds. But there's also a quarantine pests regulation in place, and regulated non-quarantine pest regulation in place, at least in the EU. But for quarantine pests, I think there is also a global agreement. But of course, there are some crops that are not traded around the globe, but more like regional trade. Regional trade is there, so quarantine pest status can also be just a regional indicated. With seeds, phytosanitary passports are also something that is provided with the seeds that are sold or traded traveling around the globe, to ensure that all test information and tracing of all the the information that on the processing of the seeds is available. And of course, import documents are also in place for seeds. So there's quite a lot of well, quite a big regulatory framework, but the importance, of course, is very big, since outbreaks can lead to very big issues in different countries, and you want to prevent those, of course. So one of the organizations that helps with this is ISTA, the International Seed Testing Association. And I thought that this is a good moment to also introduce ISTA, since Seed Health is one part of the International Seed Testing Association. Well, it's an international non-profit association for seed testing laboratories, but also for individual seed professionals. And ISTA operates under the governance of the governments of member countries and distinct economies. Well, then the objectives of ISTA, of course, is that they develop and adapt and publish standard procedures for sampling, seed testing, and promote uniform application of these procedures for evaluations of seeds moving in international trade. And another objective is to promote research in all areas of seed science and technology, and there comes our relation with ISSS, of course, as well. Well, here in the below you see this nice quadrant that actually all summarizes the different areas that we're working in: the dissemination of knowledge, the international standardization of laboratory results, and we strive for international standardization of methodologies, of course, and standardization of reporting of results. And we support that with documents and layouts and standard forms supporting seed rates. So how is ISTA organized? Well, of course, it consists of its members, but it has an elected executive committee with a president and a vice president, and also an intermediate past president. And there in the executive committee, there are also a number of committee members that are in this, that are the members at large, and help in maintaining the objectives and of ISTA and keep them in place. Well, the Secretary General Andreas is there, of course, supporting this all, and he has a secretariat that helps him out and helps ISTA out with all kinds of different stuff, like organizing and this webinar, but also helping with publications and organization of certain tests. It has it also contains technical committees. There are 17 technical committees at this moment, with 2 working groups in total. And one of this technical committees is actually the one I'm chairing, and that's the Seed Health Committee, where I will focus on more in the coming slides. So the ISTA Seed Health Committee consists of 50 committee members from all over the globe, reaching from New Zealand to Canada, from China to the Netherlands, for instance. And its core business is the development of new detection methods and updating of current methods, also known as rules, which are presented on the ISTA website under the tab of the Seed Health Committee. But it also reviews methods validations, according, of course, to the validation rules and guidelines set for seed health by ISTA. It helps in organizing proficiency tests to monitor accredited labs, but also to monitor whether the rules that are currently present on the website are still still working as they should. It's a sort of a check, of course, and it executes technical audits for accredited labs. So committee members are visiting accredited labs to check and audit their accredited methods. And this all comes with this nice diagram, which I think summarizes it nicely. And I think afterwards the sharing of the slide deck will be done, and you can go through it yourself. Then next to actually the real technical part, there's also, of course, active collaborations with other associations, and like ISSS, of course. And there's also a purpose of educating other scientists, and we're doing that via the organization of workshops within ISTA and the participation via webinars. Then there's something that's very exciting actually, and that's also there's a CTELF handbook, and the publishing of this ISTA CTELF handbook will be coming soon, which is very nice, and is one of the handbooks that is going to be present on the ISTA website, and for you to order, I assume, next to the other handbooks of the other committees that are already there. Then there's also another aspect that the CTELF committee participates in, and that's in the ISTA special projects. These are funded separately by ISTA and not directly linked to committees itself, but under the governance of committees working together. And two of them we're currently are involved in is in the ISTA reference PES lists and the pathogen image collection database, which I will come back to later in the presentation. So then on the proficiency testing and the rules. So currently there are 44 rules present on the ISTA website, and there are more coming in the coming year, I can tell you. And these are monitored via proficiency testing, and there's a PT program within the committee that we try to every couple of years, and I don't pin it on 2, 3, or 4, 5 years, but let's say within every 5 years, every rule will have a PT organized. And when do you need a proficiency test? Of course, well, there are different reasons for being a participant to proficiency testing, and that's first, of course, if you're having your own method in house for a certain pest where a rule exists for, and ISTA rule exists for, then you might apply for accreditation. And before you are accredited, it's good to already be active and try to check your method with the proficiency test. But if you want to be ISTA accredited, then of course that is really necessary that you do a proficiency test participation. It helps you check your own performance, of course, of the method. And for already accredited laboratories, it is mandatory to participate in proficiency tests. Well, proficiency tests are organized with, of course, then accredited laboratories, but also non-accredited laboratories are taking part of it. And standardly, we use a scoring of A, B, and C rating, and a B and P rating, which actually says that you're not doing a very good job, actually. A being the best, of course. And scoring of a PT report will be presented to the participants, and also uploaded onto the website for later reference as well. So it's organized by the CTELF committee members or via the CTELF committee members using the CTELF committee proficiency test guidelines, which you can find underneath, on the link, which is here presented in the slide deck as well. Then on the development and the validation of new methods, which is very important, there are two questions actually that you should ask yourself. And well, is the method that I use a good method, of course? And secondly, and more important actually, yeah, what is a good method actually? Well, to check and answer these two questions, there's a validation rules that you should refer of should use, and in there are 7 performance characteristics which you should go and check via experiments to see whether you have a good method in your hands. And well, we'll go through those. And well, the first one is the analytical sensitivity, actually the determination of the limit of detection of your test. See at what concentration of the pest you can still detect it. What is the lowest detection rate? Then, of course, is your test specific enough? So check it with a target pest, different isolates from your target pests where you want to use your test for, but also include nearly over close relatives of this target pest, but that you don't want to detect, or lookalikes in example for dilution plating or morphology studies that you can detection you can have within your protocol in the end. Then there's selectivity. Well, that's important to check whether detection of your pathogen at almost the limit of detection, a little bit higher concentration, is possible in different types of seeds. So that can be in example for brassica seeds, it can be Brassica napus and Brassica oleracea, of course, as different types if you want to test your pest in brassicas as a general. But also, if you just want to test it in tomato seeds, check a couple of different tomato batches, since you have, of course, beef tomatoes and cocktail tomatoes, which in size differ quite a bit, and also might give you a different extract with chemical compounds or other biological compounds in it, which can affect the detection. So it's necessary to do. Well, the robustness, of course, of your method. So if you have small variations in your tests, like you're using centrifuge A and centrifuge B, which are not of the same brand, that we still have the same result. Things like that you can think of. Then there are 3 actually close related ones. Repeatability and reproducibility actually are very important, of course, since it can work on one day, but it should work on the day after as well. Well, you should be able to repeat it on the same equipment and using the same conditions, and then it's repeated, or reproduce it with using it testing your method via different technicians, for instance, or using a different batch of chemicals. And these together, actually the data that comes out of it, we currently use for statistical analysis on the amount of false negative diagnostic sensitivity calculation, and also for the false positive rate at the diagnostic specificity calculation, which are set in the validation plan shared prior to starting the validation. Then on the special projects, so there are the first one, and I referred to it already earlier, is the ISTA reference pest list. There is an annotated list of seed-borne diseases, which was published by Richardson in the 1990s. It consists of about 1,300 pests and 385 plant species. But since no updates were done after 1990, the initiative started like 5 years or 6 years ago by ISTA to create a reference pest list, and updating actually the foundings of Richardson with reviewing articles. And this is done for non-vegetable species, since the vegetable the vegetable pest list, reference pest list, is actually organized via ISF, and in agreement we do in this project cover 15 non-vegetable species from 25 botanical families. Current day, and actually there is today launched the version 12 of the reference pest list, so it's already outdated. Sorry for that. And also the accurate number of 27 hosts is now up to 34, if I'm correct, with addition of three seats to the pest lists, and you can find this on the website as well. And this is promoted quite big every in different meetings, since we think it's very important to have an accurate list on this on the different pests on seeds of crops. So then the second project, which is currently running, and you will see being published by July, as the planning is now, is on a lab-based image collection, a database, which actually is going to serve as a training tool. That was actually one of the objectives, is that for people, next generation seed pathologists or seed pathologists working on new pathogens, that they can search through this database and morphologically see how pathogens look like on growing media or on blotter. And we're also going in a later stage integrate pictures of some plant symptoms as well from bioassays actually. So this will of course help to get the knowledge spread on how things look like, but there are some very important things that we think are under the radar and are not always present in articles. Is that sometimes potato dextrose agar is used, but due to the fact that a manufacturer changes an ingredient, the appearance of pathogens can on this media can be very different actually, and it is very important to know what these different variants look like, and by sharing this with each other, the knowledge exchange widens the bridge of getting good tests in place for detection. And well, this is a project that this database is currently going to be filled, but also we hope to have the submission of new images from scientists around the globe to help and enlarge and make this database growing. And this will be presented as well on the ISTA meeting in Cambridge in the beginning of July. And from that point on, we're also going to launch it so that it is accessible for everyone to check and also help to grow it. With that, I referred already that the next meeting will be of the ISTA will be our annual meeting in Cambridge, celebrating our 100th years of ISTA. Which was founded in Cambridge as well, so that's the reason why we have this nice location. Well, next to that, there are two links, one again to the proficiency testing and also to the events, where you can find the announcements of workshops, not only for related to seed health, but also, for instance, for sampling and germination or visioning projects. And with that, I like to inform you that we're together with the Seed Health Committee are going to planning to organize a webinar early next year for statistics in seed health testing. So how do you deal with comparative test results? And we are also trying, and the date is not clear yet, to organize a workshop for seed health as well, a theoretical workshop it will be, but in India with that. Then the last slide gives is there to thank you all for listening to me, and I will be glad to answer questions if they are there during the meeting if there's time, and otherwise we'll answer them afterwards. Back to you, Andreas.
Thank you very much, Ruud. And you were right, there are now 34 host species on the pest list. That is correct. And as you said, it changed today. That shows that this pest list is a very vivid and working tool, and it is used by a lot of stakeholders of the association. You saw that there was a link to this seed health imaging website. If you would use that now, it would not work yet. As Ruud said, it will be working probably sometime in July. The committee and the group is working very hard on that, and I thank you. If you want to have more information, I would advise you to go to the ISTA website and also register for the newsletter, then you get the newest information. And the proficiency test participation is free for all members, and non-members can go as well, but members have the priority to be taking part if there are not enough samples, which can be the case for seed health, because that could be quite limited in the total number of available samples. There are other benefits for members, but you can find that all on the ISTA website. Thank you very much, Ruud. I have a quick question maybe for you, only from my side. Could you explain how to join the Seed Health Committee if people are interested?
Yeah, of course, Andreas. So there's on the ISTA website, there's a tab that directs you to becoming how to become a member of the of any committee actually, and there is an application form there that you need to fill in, and that will be submitted to the Seed Health Committee chair. And when there's availability or there is a necessity for the committee for certain expertise, then there will be a voting done within the committee for somebody entering the committee, yes or no.
Thank you very much for that. I would welcome now Luna on the panel as well. Maybe you can open your camera that everybody could see you, Luna. Thank you very much, and thank you that you managed to be here with us. I think you're still muted. I would like to. Yes, we can. I would like to quickly introduce you to the audience. Luna Collard-Centenac. I hope I pronounced that correctly. You're a PhD student at the Research Institute of Horticulture and Seeds, IRHS Laboratory. and you graduated at l'École Normale Supérieure in Paris, and how you hold a master in microbiology, ecology, and modeling. And Luna is particularly interested in the interaction between plants, their microbiome, and their ecosystem under stress. That is always very interesting. I remember that a lot of my colleagues during PhD time also looked at stress situations for plants. And during your thesis, you investigated the impact of seed microbiota on seed germination, seedling emerging, and resistance to phytopathogens. And you will give your presentation on uncovering the seed microbiota diversity and its role for plant health using synthetic ecology. So the floor is yours. You may share your screen, and I would like to remind everybody there's 1 or 2 questions in the question box to put your questions there, and we will answer them together at the end of the webinar if we still have time. Thank you very much. Luna, the floor is yours. Thank you.
Thank you. Do you see my screen?
Not yet. We see you, but not the screen yet.
Can you tell me when you see it?
I will.
Okay.
Now we can, and everything looks very good, Luna.
Thank you. So thanks for inviting me and for the nice introduction. So as you said, I will present you some of my team and my own work on seed microbiota and its role for seedling health. Okay. Okay, when you think of seeds, you might imagine something like that, the seed structure, but few of us would think to add the seed microbiota to this picture. So, however, the seed microbiota is always present on the seeds, either on the surface of the seeds for the epiphyte or inside the seeds for the endophytic community. It consists of bacteria, fungi, archaea, viruses, and as I said, it is always present with the seed, even on surface sterilized seeds. As Ruud said before, some of the seed microbiota is pathogenic. It is responsible for numerous diseases on seed and seedling, including dumping off. Indeed, seeds are carrier for more than 200 different pests. I think my figure is not up to date, but you get the idea. So it's fungi, bacteria, viruses that are pathogenic for the seeds and the seedlings. But while pathogens are there are also neutral and beneficial strains on the seeds. Okay, how do we study the seed microbiota? To study the seed microbiota, we soak the seeds in water, let them macerate a bit, then we sample this microbiota suspension once the endophytic and epiphytic communities are suspended in the water. And from it, we can extract the DNA from the microorganism cells. We can represent the DNA just like that. What we are interested in is target sequences that will allow us to identify the microorganism's identity. So we will amplify those target sequences with primers, so just those target sequences. And as we've got many different microorganisms, we will have many different variations of those target sequences. They are interesting because they are quite reliable. They are shared across all bacteria and all fungi. It's different target sequences for bacteria and fungi, and with only small variations in the sequence that allow us to use it as a barcode to get the identity of the microorganism. So once we've got that, we do a second amplification with tags according to the seed sample, and we sequence those. So those are PCR processes, just as you know from the COVID period. Okay, so We sequence the molecules, and what we get now are files with linear sequences of letters that represent the sequences. Informatically, we associate them by tags, so by seed samples, remove the primers, filter the sequences based on the quality of the sequences, and associate sequences that are identical to get a number also. Once we've got all that, so we've got ASVs, it's amplicon sequence variants, we can identify the microorganism it comes from a library by comparison with libraries. Okay, so this technique, it's called metabarcoding. It's used to study the microbiota of different plant compartments, including the rhizosphere and the phyllosphere. But for seed microbiota, the interest on seed microbiota is quite recent. Here you've got the cumulated number of articles published on the different plant compartments microbiota. And you see that the seed microbiota, the number of studies on seed microbiota is really lagging behind those on rhizosphere or on whole plant microbiota. There are a few reasons for this, including the fact that the seed microbiota has a really low biomass, so we have to pool seeds to study their microbiota, and in doing so, we lose some information. Also, when we extract the DNA, we also extract plant DNA, so again, we lose some information. But I think the main reason is that historically, the focus has been mainly on pathogens, and only now do we interest ourselves with the whole seed microbiota. And why is it important to not only study pathogens? So I told you seeds always harbor some microbiota. This microbiota represents the first plant inoculum during the plant's life cycle. So it's the first microbiota, and our hypothesis is that it plays a role during the first developmental stages of the plant, so during germination and emergence. So maybe some bad microbiota could prevent the seed from germinating or lead to abnormal seedlings, while some good microbiota could help the plants to germinate and emerge and give a normal seedling, and also protect it against phytopathogens. So that raises a few questions. First, who is there on the seed? What is the first seed, the seed native microbiota? And then, does it impact the seedling phenotype? The seed microbiota, does it impact the seedling phenotype? And if so, can we use it to improve the seed quality or the seedling phenotype? And if we do, how is this seed microbiota transmitted to the plant at later developmental stages? So first, who is there? I will present you some results of a meta-analysis that was done in my team. This meta-analysis covers 63 studies over 28 countries and for 50 plant species. One of its main results is that for each seed sample, in median, we find 48 different bacterial taxa and 42 different fungal taxa. So it means if we look at the identity of all the microorganisms that are on the seed or in the seeds, we've got 100 different taxa, half bacterial, half fungal. And if we look at all those identities for all the seeds batches of all plant species that were considered in this study, we've got over 8,000 different bacterial taxa and 2,000 different fungal taxa. It's quite huge. Now, if we look at the distribution of those taxa, here you've got the number of different taxa, bacterial or fungal here, found in this number of different plant species on seeds, always. So what you can see is that for bacteria and fungi, more than half the taxa found are found only in one plant species seeds. But there is a few taxa that are found across different plant species. So let's have a look at those. Those 13 bacterial taxa and 16 fungal taxa represent the core microbiota of seeds. It means that they are the fraction of microbiota that is found, well, either in a specified host, regardless of the environmental condition, or here for us, in a specific environment, regardless of the host species. And they are stable in terms of relative composition and across time. So here, for those 13 bacteria and 16 fungi that are found in many different plant species, who are there? For bacteria, it's mainly Pantoria, Pseudomonas, Sphingommonas, and Rhizobium. For fungi, it's Cladosporium, Capnoteres, Alternaria, and Vishnia cosina. But that does not tell us about how many, which proportion of the microbiota they represent. So let's have a look at that. The 13 most prevalent bacterial taxa, so those that are found across different plant species, represent up to 41% of the relative abundance of the bacterial community. And the 16 most prevalent fungal taxa represent 47% of the relative abundance. It's enormous. So to sum up that a bit, the seed microbiota is composed partly of plant-specific taxa, but half of it is core microbiota. So it's both prevalent and predominant taxa that are shared across plant species, and that makes up most than half the microbiota. And in total, those represent 10,000 of different taxa, both bacterial and fungi. Okay, now that we know a bit more about who is there, we would like to see if they impact the seedling phenotype and if we can use this rule. To do so, well, this is one of the questions of the Succeed project I worked with, and its aim is to stop the use of pesticides on seeds. As you may know, the seeds are often covered in pesticides, mostly fungicides, but also insecticides. And in accordance with societal concern, there are projects that aims to find alternatives to those pesticides using, for example, seed microbiota. So for the Succeed project, it focuses on 4 different crop species, and my work is only on the common bean species. So I will present you some results. We work with 8 different varieties of common bean Phaseolus vulgaris. And for this work, we obtained the seeds from the FNAM experimental station. So the plants were grown at the same time and in the same field. And then we characterized the quality of those seeds of these 8 varieties. The seed quality can be defined as the seed ability to germinate and emerge quickly and uniformly once sown. And it means that for a batch of seeds, we want all of them to germinate quickly, simultaneously, in various conditions, and against various stresses. We also want them to produce normal seedlings that will be more productive. It's quite visible with common bean seedlings because we've got some normal seedlings, but also a lot of abnormal seedlings that have either smaller leaves or are deformed in the stems or their roots. Okay, for our 8 varieties, we characterize the quality with the germination. So here you've got the cumulative germination percentage of our different varieties in different colors through times. And we also characterize the emergence percentage of our varieties at 7 days after sowing in potting soil. For a first analysis, we can group our varieties into 2 groups, one of high emergence percentage varieties and one of low emergence percentage. So the blue for the low emergence percentage varieties and the varieties in yellow-red with high emergence percentage. So we've got this characterization. Now we would like to link it to the seed microbiota. So we first characterize it. Here you've got the different common bean varieties, and for each one for different samples, batch of seeds, you got a relative abundance of the microbial taxa. Here's the fungi first. I will show you the bacterial just after. So we find back our core microbiota taxa, so Cladosporium, Alternaria, and Stemphilum for the fungal community. Here, all taxa comes from 23 families, and we've got from 25 to 80 different fungal taxa in our seed batches. Here, the plant variety explains for, so you see differences. Sorry, you see differences between the relative abundance of the microbial community in different plant varieties, and the plant variety factors explain for up to 78% of the fungal community variation. Now for the bacterial community, again, we find back our core microbiota, so Panthoea, Pseudomonas, Sphingomonas. Here we've got between 50 and 250 different bacterial taxa coming from 19 families, and the plant variety factor explains for 42% of the bacterial community variation. A small parenthesis here. So we characterized the microbiota for seed batches. Now let's have a small look at the single seed. If we take only one seed, in average, we find that it harbors only 8 different taxas, both bacterial and fungal, and the community is dominated by only one taxa. And if we count the number of cells, we've got only 100 colony forming units. So for one single seed, we have a very low diversity and low abundancy microbiota. Back to our 8 common bean varieties and the link between the seed quality on our seedling phenotype and the microbiota. So here we would like to link the seed microbiota with the seed quality, sorry. So to do so, we have those 2 groups of varieties, with high emergence percentage or low emergence percentage, and we search which taxa are more associated with one of the group. So here for bacteria only, you've got the high emergence percentage varieties here, the low emergence percentage common bean variety here. And we can see that some bacterial taxa are mainly found in high emergence percentage variety, while for others, they are only found in low emergence percentage varieties. So that is a correlation. Now we would like to study the causality. Those taxa directly impact the seedling emergence. And to do so, we will use synthetic community approaches. So the aim is to study the link between the seed microbiota and the impact on the seedling phenotype. So to do so, we use synthetic communities approaches. That means that we will design some association of microorganisms that mimic the native microbiota, but in a simplified manner. And we will use it to inoculate the seeds, to replace the former seed microbiota. Then we will sow those seeds in non-sterile soil, let them grow, and study the phenotype of the seedling at 7 days after sowing. To design those synthetic communities, there are a few steps to proceed. Okay, just before, sorry. So we do that with many different synthetic communities in order to link the presence of some strains or some associations of strains with the seedling phenotype, either normal, above, abnormal, in the middle, or enhanced in the bottom. Okay, to design the synthetic communities, we've got a few steps to follow. First, we need to isolate microorganisms from the plant microbiota. We need to select taxa that might be interesting, that can enhance the seedling phenotype, for example, and we need to efficiently inoculate the seeds. So The first part, the isolation and stocking of plant microorganisms, was done in the ISTA-ISSS lab for the common bean from those 8 varieties I presented you. And so the strains were isolated from seed batches and seedlings. The microorganisms were identified, and they are now stocked at the CFBP collection for bacteria and in another library for the fungi. We can use this collection. So for bacteria only, I will present you for bacteria only. We have a huge collection of bacteria isolated from the common bean, more than 1,000 taxa. And we select a few strains among those. Those are the one depicted with the red dots here. We select randomly some strains that we will use to design our syncops. So we will inoculate them. So first we need to screen them to see if they don't have bad effects on the seedling phenotypes and to select those that improve the seedling phenotypes. To do that, we inoculate them on the seeds and we look at the seedling emergence percentage. So here in gray is the control emergence percentage here. And you see that from the 40 different strains that we selected, most seem to improve the emergence percentage, but 3 we will exclude because they decrease the emergence percentage. Those are 1 Pseudomonas syringae and 2 Pseudomonas viridiflava. Okay, so we've got all those strains that were isolated that we can use. We'll screen them. And now we need to know if we can efficiently engineer the seed microbiota with consortia, associations of those strains. So we can check it by looking at the relative abundance of the strains we chose in the inocula. So the solution, the suspension we use to inoculate the seeds. Then on the seed, so here each bar represents 1 seed batch. And what you can see is that we find back our taxa, the taxa that we are inoculated, we find them back on the seeds and in the same relative abundance. So that's very good. We efficiently monitored the seeds microbiota. We can also look at what we find up to the seedling developmental stage. Okay, so that's the same principle. And so for different synthetic communities, you can see here that in each case, we efficiently engineered the seed microbiota. The taxa that we inoculated represents more than 80% of the total microbial abundance. And the transmission to seedlings depends on the identity of the strains we used. So one color represents 1 taxa in all those graphs. So the seed microbiota is efficiently engineered by the synthetic community inoculation, and the transmission to the seedling depends on the composition. Now we want to look at the effects on the phenotype of the inoculation of some synthetic communities. So here is the emergence percentage of our seedlings, either the control condition or for inoculated seeds with different synthetic communities. You can see that for all synthetic communities, it seems to decrease the emergence percentage of the seedlings, which is not what we expected. But now if we look at the ratio between normal seedlings among all emerged seedlings, you can see that compared to the control here, all synthetic community inoculation improved this ratio. So our synthetic community seems to have a beneficial effect that is yet to test on different media and conditions. Yeah, so here the inoculation decreased the abnormal seedling percentage from 30% to 10% depending on the synthetic community. It's really good. And finally, if we use these solutions of synthetic community, we need to know up to what developmental stage they will be transmitted and if they have impact on the plants at later stages. So to do so, we also inoculate our seeds with synthetic communities. It's the same kind of graph as before, so you know how to read it by now. Here, we decomposed the analysis by seedling compartments, by plant compartments. So compared to previously, we also have the information now that the taxa we inoculated are mainly found in the leaves, in the aerial part of the seedling. Now let's look at further developmental stages. So first, trifoliate, floral bud, fresh seeds, and mature seeds plants. So always for the common bean. You can see that we do not find our taxa back at later developmental stages. So the seed microbiota is progressively replaced during the plant's life cycle. So it is a really interesting solution to use, those synthetic community, but their action might be time targeted. Which brings me to my conclusion. So we wanted to know a bit more about the seed microbiota, the good and the bad, and how to use it. So we first wondered who was there. So for individual seeds, they have a low abundancy, low diversity microbiota. Some of the taxa are shared across plant species. It constitutes the core microbiota, and some of them are plant-specific taxa. Also, the seed microbiota constitutes the primary inoculum, mostly for above ground compartments, so for the leaves of the seedlings, and it has a role in seedling emergence. It is also progressively replaced during the plant life cycle. So seeds are a promising vector of microbial solutions, either for enhancing the seedling phenotype or to protect them against phytopathogens. And with that, I would like to thank my supervisor, my PhD supervisor, the two teams I work in, MRC's team and the SMS team, and our partners, so the FNAMs for providing the seeds, the GVS that did a lot of mature seed characterization, Plato-Anan, where we sequenced our microbiota communities, the Phenotic platform, where we phenotyped our seedlings, and the Succeed project, which granted me my PhD project. And thank you all for your attention. If you've got questions, I'm available to answer.
Thank you very much, Luna, as well. Quick question from my side. Are there commercial applications available for what you presented?
Okay, so for now, there are already a few suspension solutions that can be used that are commercialized, but mostly they are single seed solutions. And we know that the microbiota, the interaction inside the microbiota are really important for the reproducibility of the effects on the seed quality, the seedling phenotype, and the plant health. So it yet has to be developed.
Okay, thank you very much. I may go through the questions. Maybe we start with you, Luna, on that. That is maybe the easier way. There was a question coming in. As per the information presented on seed microbiota may vary with the soil health, previous crop history, and so on and so on. Some beneficial microbiota like Rhizostopium, Pseudomonas 2 are found on common bean. but seeds are treated with fungicide, whether to coat with plant protection chemicals or not? Difficult question.
Okay, so about the variation between seed microbiota depending on the plant, well, the soil or the plants, yes, it varies, but so I showed you the definition of the core microbiota. There are strains that are always found in the seeds, and depending on the condition, yet the seed recruits some of its microbiota during its developmental stages up to the seedling stage, and we don't know a lot about the transmission from the mother plant to the seed, but of course it also impacts the seed microbiota assembly. About the second part, can you please repeat? Sorry, I did not understand.
Let me find that question again. That was a treatment with fungicides, as far as I remember. Or any treatments, here we have it. Whether to coat with plant protection chemicals or not.
Whether the
If it is, if the coating should be done with plant protection chemicals or not. Maybe Ruth, you can help out.
Well, of course, there's something to say about the different rhizospheres due to the fact that there's different soil types where you plant your seeds in. Can you prevent that with certain coatings? Well, you still have an effect, of course, of different microbiota in the rhizosphere that reacts with or attacks actually your seed germinating, and then, well, as your microbiota present on your seed has a reaction in the different soil types, also does the soil type have a different effect on additional coatings. So if you would yeah, if that solves your problem, I am not really sure about that, and that's it's difficult. Soil studies are really difficult, and the impact of soil is something unpredictable actually.
Yeah, what I may be able to add, not on the seed side here, that really differs to the soil, to the situation in the soil as well. And when you talk about bioenhancement products or whatever, they are mostly designed for a specific soil, for a specific environment, and very, very similar here for treating seed, and because that has to be done for a specific environment as well. If you use, if you don't use pesticides, which we want to reduce in the moment. Luna, thanks for the nice presentation. Very great job. One question back to slide 17. You showed us that microbiota diversity in seedlings vary from the one in the initial inoculum. Now I have to, it's not very big here. Maybe I can make it a little bit bigger for me to read. But that doesn't look like. No. No. From the microbiota and seedling varied from the one in the initial inoculum on seed. The experiment was done using non-sterilized soil for sowing the seeds. Did you have a look to the soil communicated before sowing, after sowing, at the time of your investigation? The seedling associated communities. What about a possible impact of the soil on the changes in the seed to seedling evolution of microbiota? I think goes into similar direction.
Yeah, so about the, well, first about the taxa we choose for the synthetic community. So there were a few pathogens, that was the Pseudomonas I showed you, but most of the strains we randomly selected were not pathogenic. And we tested their beneficial effects in community, so in the synthetic community tests. About the effect of soil microbiota, of course it has an effect, but why we say that the seed microbiota represents the first inoculum of the plant is also that they have the priority effect. So they are already there, present on the seed, so they might have a competitive advantage against the microbiota that will try to colonize the plant from the soil. And I don't know if that answers the question.
I hope. Let me go to Ruth. One of the first questions which came in was, I would like to know which type of quick tests are being used to detect pathogens in seeds and nerves.
Well, a quick. There are a lot of different ways of detection, of course, and quick. Yeah, you can consider quick as within 6 days or within 1 day. There are a lot of molecular techniques currently available, assays available, which are really specific. Only the difficulty there is that detect your DNA, and it can be DNA can be of alive pests, but also a residue of dead pests can still be present. So it gives you an indication. Only if something is not present, then you won't detect anything. So that's quick and dirty tests that you can do within a day. Otherwise, the dilution plating for bacteria is, for instance, a very good technique, and it takes you about 5 to 6 days, which really shows you the on semi-selective media, the growth of the target bacteria. Yeah, then you know when it's present that it is alive. And confirmation is then done by pathogenicity testing, which can be done on plants, or either by a molecular test. And then the molecular test gives you an definite answer. For viruses, ELISAs are very common, of course. That's also quite. It's an easy test, and it takes you about 1 or 2 days.
Quick answer to that. Can we conduct a seed health test after seed treatment?
Well, that depends, of course, what kind of seed treatment you have. We know that certain chemical treatments do interfere with DNA isolation, but others don't. And with molecular testing, of course, then well, that DNA will be prevalent, so detection is there. And yeah, it may interfere with growth conditions of bacteria with dilution plating, for instance, as well. Yeah.
For the previous question, there was a second question coming back. Quick, I say, in the same day.
In the same day on seeds.
Yeah.
Well, you can, of course, and well, we do not validate those, but there's a possibility, of course, for the dipstick method. So there are antibody strips available for different pests. So if you create a seed extract, then you can just dip it, and it works like a pregnancy test. It gives you a plus or a negative. But we don't consider those as really reliable.
Yeah, the quick and the more dirty is the more unreliable
Yeah, and next to that, Andreas, just pops in my mind, there are LAMP assays available. Also molecular assays, so guaranteed to the door, but those take you about an hour to get your result. But then you're target specifically looking, and yeah, you can nowadays these devices go up to 24 samples, I think. So it's not really high throughput. That's
Okay.
the disadvantage.
Luna, do you pathogen tests with Pseudomonas isolates?
I did not work specifically on Pseudomonas, so I don't have specific tests for it. For all our strains, either alone or in synthetic communities, our main test was to inoculate them on the seeds and then look at the seedling phenotype at 7 days after sowing. So it's not a specific test for Pseudomonas.
Okay, yeah.
I actually have a question for Luna, if there's time, Andreas.
You go ahead, yes.
So these synthetic communities that you're talking about, I assume that they're not really easy to prepare and reproduce even. So my question actually is, did you, the data that you showed, is that from one experiment done on multiple seeds sown, or is it done over time?
For the results, well, the preparation of the synthetic community, it's quite easy once you've got the trick. Of course, there are slight variations in the relative abundance between experiments. So each time we characterize both the inoculum relative abundance, well, the relative abundance of the taxa in the inoculum on the inoculated seeds. So that is for each experiment. And here the results I presented you of the synthetic community inoculation. We did it once at the same time for all synthetic communities, and we reproduced it later with a few of those synthetic communities that showed a higher impact on the seedling phenotype. But we still have to test it in different soils and different temperature conditions, well, to see if the effect is reproducible.
Okay, thank you.
And I've got, due to the time, a last question, which maybe go to both of you. Does practical more seed soaking better overnight, or just 2 or 3 hours with agitation is enough?
Well,
Good question.
I can give an answer to that. That really depends on the type of seeds that you have. If you have open field seeds, then I would say that shorter time will prevent saprophytes from growing in the meantime. Whether you have seeds that are from greenhouse crops and from inside fruit, so not directly open to the environment, these are better to do overnight soaking for the extraction, because you really, yeah, well, you think the reason of the idea is that the pests are entering the seeds via the fluid and the sap streams that are in the plant. So they're more internally expected, and then a bigger incubation time is better.
Thank you to both of you. We will have a few number of, and not a lot, but some more questions. You will all get answers. We will forward them to the panelists, and you'll get answers to that. So don't worry if your questions were not answered during the session. But we try to stick to the time, as we can also see that at the end of the session, people are leaving and leaving and leaving. So that is important. I would like to thank you again for that. Very nice presentations, very well done. And remind everybody that the presentations, and I posted it 4 times in the chat, are available also online, and you can have a look at the presentations. And if you have questions, probably both will be able to answer them even at a later stage. And that was a very successful webinar again, together with ISSS and ISTA in that case. Thank you very much. Julia, you want to say a few words at the end as well?
No, just have a nice day, everybody, and a big thank you to the speakers.
Thank you very much, and see you. Thank you. Bye. Goodbye.
Okay. Bye bye. Thank you.
Thank you very much. Bye.